So first, I will give you a brief introduction on the platform and our assay principle. Then we will discuss the sample characterization capabilities that we offer. Then I will give you some examples of the assays offered with this platform, including standard of care and targeted therapies, as well as immunotherapy, assessment in native tumor microenvironment, and immunotherapy assessment in enhanced tumor microenvironment. Finally, we will look through the whole sample journey and summarize the platform insights. As you're probably well aware of, there is a huge translational gap in vitro and in vivo models for oncology. And this contributes to the very poor clinical success rate of the oncology drugs for the patients. We believe that to improve the clinical success rate, we need to find the better translational models that represent the molecular and genetic complexity of human tumors and better represent human tumors biology. We believe that this is feasible through the models that represent a native tumor microenvironment and can help to get a more accurate insights beyond the general cell viability assessments. Of course, the native tumor microenvironment includes endogenous immune cells, and this is especially relevant for evaluation of immuno-oncology drugs. Therefore, we believe that these models can help you make better decisions into progressing the drug candidates to the clinic. Our models are based on physiologically relevant cultures that are derived from the fresh patient tissue, containing all endogenous cells of the tumor microenvironment, including immune cells, fibroblasts, and other stromal components. We work in a high-throughput manner in 3D with 3 to 4 well-placed imaging platform with cultures exposed in 3D in a hydrogel. Our readouts are phenotypic based, and we have a proprietary high-content image analysis platform to measure these readouts. So we start with the fresh tissue that comes to our laboratory in Leiden within 24 hours after collection from the hospital, from the patient. And then we use very gentle mechanical processing of this tissue to generate tumor clusters of approximately 100 to 150 micrometers in size, which are then mixed with the hydrogel and seeded in a 3 to 4 well plate format. Together with these clusters, we preserve all endogenous cells, including immune cells, fibroblasts, and other stromal components. The cultures are then exposed to the drugs, and we keep the cultures for about 5 to 7 days. Afterwards, we fix and stain the cultures and do the imaging and optical sectioning in 3D. And then our proprietary image analysis platform performs the image analysis, and we can extract multiple phenotypic readouts from these cultures. The example of the readouts are number, size, and shape of the tumoroids, cells and nuclei. We can also analyze different cell subpopulations and proliferation of immune cells, for instance. In addition to our high-content imaging readouts, we have also available immunohistochemistry, characterization of the samples, sequencing, flow cytometry, and we also collect supernatants from these cultures to analyze various cytokines. On this slide, you can see an example, some examples of the 3D ex vivo cultures that we work with. We have experience with ovarian, breast cancer, bladder cancer, lung cancer, and cervical cancer. We also have experience with mesothelioma. And when we analyzed the histology of our ex vivo 3D cultures and compared them to original patient tissue, we found a very good matching between the biology of our 3D ex vivo cultures and the matching patient tissue. And this matching was also confirmed by the hospital pathologists. On this slide, you can see really the complexity of our tissue compared to the tissue that have been passaged for a couple of times. So if we start on the left here, this is how the tissue looks like during the isolation you can observe some tumor clusters and some single cells around them. Then when the tissue is seeded in a 3D environment and we run our assay, you can see a very complex biology present with the tumor clusters, fibroblast-like cells, and immune cells around them. However, if we passage this tissue a couple of times, even in 3D, we observe that this complexity of tumor microenvironment disappears, and at some point we only have tumor clusters present. In addition to immunohistochemistry, we can characterize samples with FACS, for example. On this slide, you can see a FACS analysis of single cell fraction isolated from non-small cell lung cancer sample. And these single cells were mostly immune cells, CD45 positive lymphocytes, and 70% of them were CD3 positive T cells. We can also characterize samples by immunofluorescence. And on the image on the right, you can see the staining of non-small cell lung cancer ex vivo tumor tissue with EpCAM in green, marking the tumor clusters, and CD45 positive immune cells in cyan. Now, if we move to the assay itself, what we can analyze in these cultures. Here you can see an example of images from an ovarian cancer sample that was exposed to different standard of care drugs and targeted therapies at various concentrations. And each image represents an entire well of a 384 well plate with the tumor clusters stained with DAPI and actin. So the more actin staining we see, the more viable are the tumor clusters, like in a non-treatment condition. And our image analysis software is able to segment these tumor clusters and analyze them, what you can see in the segmentation mask. Of course, we do all this in 3D, so every image is a 2D projection of a 3D stack. And you can see different responses of this patient sample to the different treatments. And then if we put these responses into numbers and analyze different concentrations of the treatments, we can really build IC50s and analyze the responses of this patient tissue to various drugs. On this slide, you can see a heat map of about 30 ovarian cancer patient tissues that were ranked from drug sensitive to resistant based on their responses to different standard of care and targeted therapy compounds. As you can see, part of the patient population is sensitive here on the left to most of the treatments, and the other part is resistant to most of the treatments. And the patient population in general is sensitive to some of the treatments and resistant to others, as you would expect in a real patient population. Now, in the proof of concept study, we took two different samples from this population, one that was sensitive to most of the treatments and another one that was resistant to most of the treatments, and we analyzed their genetic background. It turned out that the patient tumor that was sensitive to PARP inhibitors and to platinum drugs, presented here in blue, was a BRCA mutant tumor, and it was sensitive to these treatments, as you would expect for the patient tumor to be sensitive in the clinic. And the other patient tumor that was resistant to the treatments to PARP inhibitor and to platinum drugs was a BRCA wild-type tumor. So this assay confirms our functional readouts that correspond to the genetic background of the tumors. As I already mentioned, this platform is preserving endogenous immune cells and therefore makes an ideal platform to analyze immunotherapies. Here we can offer two different options. First, we can test immunotherapies in a native tumor microenvironment, so with the immune cells that are present endogenously in a sample. And in this case, we would have the readouts such as tumor killing and immune cell proliferation. Additionally, we can acquire a whole blood sample from the same patient isolate specific immune cell subsets or just PBMCs. And in this case, we will pre-stain them with a separate staining and mix with the sample having already some of the immune cells present, but this will give this additional boost to the tumor microenvironment. And we will have additional readouts available for these pre-stained immune cells, such as immune cell migration and infiltration into the tumoroids. When we look at the immunotherapy effects in the native tumor microenvironment, there is a typical phenotypic signature that we observe. On this example, it's ex vivo tumor tissue from non-small cell lung cancer patient, where in the non-treatment condition, we have a lot of healthy tumor clusters, as you can see here on the image on the left. And when we apply our immunostimulatory control, superantigen, that stimulates non-specific immune response, we see a decrease in the tumor clusters, in their size and in their numbers. And we also see an increase in the numbers of single cells or immune cells. The sample was also responding to a checkpoint inhibitor, ipilimumab, and we have seen a similar phenotypic response from this treatment as well, where we could see a decrease in tumor fraction and increase in the single cell fraction. So how do we analyze this quantitatively with our platform? Our bioinformatics pipeline has specific filter set. First filter set that is designated to specifically segment tumor clusters and analyze their number, shape, and the volume. And the other filter set specifically analyzes immune cell counts. On this video, you can see a comparison cells fractions increase. We can see this again on the video that red are single cells and tumor clusters are represented here in green. When we put these phenotypic readouts into numbers, we can observe this kind of data where in this sample there was a very strong response to the ACA stimulation. So we have seen a drop in the total tumoroid area, and there was also a response to a CSF1R inhibitor with, again, a drop in the total tumoroid area. However, these two drugs have a very different mechanism of action. The SCA stimulates immune-mediated killing, and CSF1R inhibitor acts through the direct tumor targeting. So then in case of SCA, we are observing this increase in single cell counts, so increase in immune cell proliferation, but we do not observe this for the CSF1R inhibitor. Also, when we analyze supernatants from these cultures, we can confirm that in this specific sample, in case of SCA, where we see an increase in immune cell proliferation and the killing of the tumor, we have also observed an increase in interferon gamma production, confirming our functional readout from the phenotypic analysis assay. Another good example of immunotherapy responses is this ovarian patient tissue, where we have seen almost complete killing of the tissue in case of treatment with ACA, as shown on the graph here, and also very strong reduction in the tumoroid area upon treatment with checkpoint inhibitor ipilimumab. And when we analyzed histology of this sample, we could detect tumor clusters, CD3 cells, and also that tumor clusters were proliferating. So the histological readout was again confirming our functional readout from the image analysis platform. Another good example of immunotherapy response in the native tumor microenvironment is this ex vivo patient tissue from non-small cell lung cancer patient that was responding to SEA and to CD3/CD28 beads by a reduction of the tumor area, but it was also responding to a panel of checkpoint inhibitors, including ipilimumab, nivolumab, and pembrolizumab. And when we analyzed the histology of this sample, we could detect live tumor clusters, CD45 positive cells or lymphocytes, and also PD-L1 positive cells that, according to the pathologist, were macrophages. So all together put into perspective, our assays in a native tumor microenvironment can show the patient tissue sensitivity to immunotherapy drugs. And what we observe in general as a trend, about 20% of the patient tissues respond, for instance, to checkpoint inhibitor treatments. And this is very similar to what is observed in the clinic. Now, if we study the effects of immunotherapy in an enhanced tumor microenvironment, what I mentioned earlier, the co-culture with autologous PBMCs, we can directly visualize the PBMCs in these cultures because they are pre-stained. Here they're visualized in green. And we see in this patient sample, this is from non-small cell lung cancer tumor, a very strong response to our immunostimulatory controls, ACA and the beads, and also strong responses to different checkpoint inhibitor drugs. In this case, we can directly measure PBMC counts and infiltrations in the tumor because they are pre-stained. Additionally, we can isolate and expand tumor-infiltrating lymphocytes from the primary samples and expand the tumor culture and put them back together in the co-culture with a matching TIL. In this case, the responses of the TIL would be slightly different than on a fresh culture because, of course, these cells were expanded. But still, in this example, we can see a strong response to SEA by the killing and a slight increase in tumor infiltration in the response to the treatment with ipilimumab. All in all, to summarize the sample journey, we receive samples fresh from the biobank within 24 hours from collection. We minimally process them to preserve tumor clusters and all resident cells, including immune cells and fibroblasts. And when the sample is ready for the seeding, there are multiple things we can do with it. First, of course, we can do our 3D high-content image analysis assay in the 384-well plate. And from this assay, very easily, we can collect supernatants and run further cytokine measurements. Additionally, we can preserve part of the sample for immunohistochemistry, FACS analysis, or sequencing. Also, we can run a parallel culture, which will be scaled up compared to our HCI assay. And in this parallel culture, we can do immunofluorescence, FACS, or RNA-seq analysis. So I hope I convinced you today that this platform offers the most physiologically relevant preclinical environment available by working with the fresh tissue with native tumor microenvironment in 3D. We can evaluate different therapies in this platform, standard of care, targeted therapies, immunotherapies, including immune checkpoint inhibitors. And we can do this in using endogenous immune cells, but also with autologous TIL or PBMCs from the patient. The platform allows to accurately measure tumor killing, immune cell proliferation, migration, and infiltration, thanks to our proprietary image analysis capabilities. And we hope that this platform can help you to gain more insights into mechanism of action of your drug and to help to progress it into the clinic. Other in vitro solutions offered by Crown Bioscience are presented here. You can see that we have an extensive collection of 3D tumoroids, of PDXO tissues, patient-derived tissues, and finally 3D ex vivo patient tissue platform that offers the most complex assays with a native tumor microenvironment, including different cell types. Thank you for your attention, and I'll be happy to answer your questions.